Dr. Tim LaSalle. Thank you so much for joining us, Tim. I've got a brief intro for you here. So since 2007, Dr. Tim LaSalle has championed his science-based hope for a regenerative food system that will mitigate climate change by carbon sequestration in place of soil carbon loss. LaSalle has served as the first CEO of the Rodale Institute, which is quite a prestigious institute doing some amazing research, the executive director of the Northwest Earth Institute, an organization dedicated to grassroots movements, and also executive director of the Allan Savory Center for Holistic Management. They're an international nonprofit whose mission is to restore and regenerate deteriorating landscapes. And he's also recently the co-founder of Cal State University Chico. They have a regenerative agriculture initiative that he's leading the charge on. He hails from the Central Coast, and you're also a member of the Central Coast chapter. Is that right, Tim?
I am.
Right.
Proudly.
Nice. So fellow CRFG member, very soil focused. I'm excited to hear what you have to share with us on soils and compost, and help us all sink more carbon into our soils while we grow our fruits. Thanks for coming.
My pleasure, Ty. I'm going to put share screen now and see if we can get this. My PowerPoint right here is what I want. And why is that not showing?
While you're setting that up, I'll do a little bit of housekeeping. So brief housekeeping using Zoom for anyone that's not familiar. We do have a chat box that you can click on at the bottom of your screen. There's a chat button. Feel free to ask questions that'll be addressed at the end of the presentation in the chat window. And you can shrink the speaker view to whatever size you want, move it around, have it just focused on Tim, as opposed to multiple members. If you want to maximize your screen real estate, there's buttons. Once he shares his screen, we'll be able to shrink.
I am not getting this shared. And you do have, looks like you do have me on sharing.
Are you seeing the green button at the bottom that says share screen?
But then when I come up, my PowerPoint is not coming up, which is really curious. Oh, here it is. I'm not seeing it. My fault. All right. I apologize. Okay, let me get it to full screen and we can begin. Ty, thank you for the introduction. I just want to say that I've had the opportunity, I'm a professor emeritus from Cal Poly from the 70s and 80s, and just went out into the world because I saw so much change in the world, particularly in agriculture, in decline and degradation, saw populations growing, understood that we really were exhausting this planet, and it changed my high input agricultural consciousness. It changed my idea of how we farmed and scientifically how I was teaching it. And it made me pause to reflect is what's going on in the world where people are innovating to do this differently. And that took me through all of the course through the Organic Center, Rodale Institute, and eventually with Howard Buffett to Africa, where I spent 4 years combining no-till farming with a biological approach. And so what I'm going to posit to us tonight is a new set of lenses for us to begin to think in terms of our relationship with earth, with our trees, with our soil, and to also begin to try and think in terms of what paradigms have we brought to those trees and that soil, and where have we sort of been misinformed in the past, and what might the new possibilities look like where we work more with nature, and what might that bring? So with that, paradigm paralysis is what we all get caught in, and we sort of see it in today's, arguments and arguing about facts, which ones are correct or wrong. But in the scientific world, Kuhn had looked at this years ago and said, our training sort of puts us in one way of thinking about something. And when that happens, it narrows our frame. it actually limits our ability often to make and find solutions, because it's sort of like the old statement, if what you have in your hand is a hammer, everything you look at looks like a nail. And that's where we look and think in terms of many of our soil scientists. They're geochemically trained, and they're thinking about the soil and its deficiencies because they've chemically analyzed it, not understanding there's a whole new paradigm that is emerging in our awareness, not emerging in our awareness, that we can work with very, very productively. So when we look at that, and for some, there we go. I don't know why that did that. Here's where we find ourselves as a planet, a species. You know, this COVID-19 came along and we thought it was going to overwhelm us or kill us, and we were worried about a recession coming. But behind that was really something we're starting to talk about, which is climate change. And then someone said, well, boy, the biggest thing is biodiversity collapse. Well, here we sit in an existential crisis, and we feel often powerless about many of these things. Because you are involved with soil and with plants, you're not only not powerless, you can help make a huge change that will make a very significant difference. And I will share that new paradigm with you tonight. This is just a little thing on water freezing that we talk about as an example of a paradigm. If I asked you what temperature does water freeze at, although those of you in Orange County see this less than we do here, but is, you know, we'd say 0 degrees centigrade or 32 degrees Fahrenheit. Actually, pure water freezes at minus 38 degrees Fahrenheit. The only reason it freezes at 32 is because there's organisms in there called Pseudomonas syringae, which helps ice form at a much warmer level. And that starts to change our paradigm of understanding. It's not really just water that's freezing. It has to have a living presence of these organisms to freeze at the temperature we're used to seeing it freeze at. It actually then helps make cloud formation. And we always think in terms of clouds coming in and rain sort of being as this real external thing, but the plants contribute to forming the raindrops and bringing the rains down, not just particulate in the air, but actually microorganisms. There are rain-making microorganisms that live in the leaves of trees. And so forests and trees and what you're planting in your backyard all help the rain come. Covering your soil does too. So we're going to put on this new set of lenses as we now think in terms of the soil in tonight's conversation, and look at it perhaps through a very, very new paradigm, and that's the lens of the microbiological world. What we are learning, and I sort of understood this when I was at Rodale, and we had these carbon numbers about how much carbon we could capture that was so much higher than what other scientists were talking about. And it really made me think in terms of, yeah, when you get a living soil, things work differently versus a dead soil. And in essence, at that time, there was sort of some work starting on the human gut biome. And what we've learned with the human gut biome is that, in essence, the microbiology that lives in our gut is going to determine a lot about our immunity capacity, how our immunological responses are. What we also find is it can cure diseases like Crohn's disease. There's a whole group that, if that microbiology is changed to be very healthy with a broad spectrum, the health greatly improves. The third thing that I think is odd, but it's very true, is that it also affects our moods. I could tell you a story about that, which we won't have time tonight, about somebody who saw his immediately change when he changed the microbiome. And sadly, you can't do it just by buying probiotic downtown. Those are too narrow of a spectrum of species to actually accomplish what they're kind of claiming to accomplish. Now, there goes my program, kind of. There we go. So we just mentioned the gut microbiome can improve our health. It can generate metabolites, which are cell energy sources, promotes health, and prevents actually pathogenic microorganisms. You know, one of the things that I think we forget about is that we have pathogens on us, in us, and it's not a matter of having them. Actually, they're probably pretty important to stimulate immune responses. But there's a balance, there's a relationship between what we would call the good ones in coordination with the bad ones. We have a microbiome in our gut, on our skin, in our lungs. And if it's healthy, we're healthy. 10%, and I want to mention this to you, this is a really curious thought, 10% of the cells in and on your body are yours. The other 90% belong to the microbiological world. And that's interesting. We're sort of carriers for a whole community. You know, it's kind of interesting. They say a dog might be the child's best friend. I would often say a very healthy garden in their backyard is a child's best friend. To be playing in, to dig in, to put their hands in their mouth from, to avoid the kinds of respiratory disorders that children raised in very clear and clean environments, quote unquote clean environments, are suffering today. There's this inoculation in the birth canal when a baby is born normally that is the microbiome that starts them off to survive well in this world. So here we are thinking in terms now of, in this new lens of looking at it, how does this fit within soil? And I'm going to talk to you tonight a little bit about a Johnson-Su bioreactor, or an inoculant that you can make in your own backyard. And what's interesting here is that what's the difference in this chestnut tree between the branch that's leafed out and the branches that are barely leafing out? And what is happening here, and one of our rare fruit tree growers here, Joe Sable, is a great grafter, and that's why I put that note in here. No, it's not that that tree was grafted. It's in fact that this farmer was spraying this inoculant on his grazing land, and some spray got up into the tree. And that branch that got sprayed leafed out a month earlier and was way ahead of the rest of the tree. So microbiology started to shift even this, when we talk about it on our skin, started to shift the health and response of that part of that tree. Also, I was doing hosting a rare fruit tree grower meeting on my place, and I was doing a little bit of this talk, and one young lady who grows quinces and jujuberries went home and made a bioreactor, and she put this on her quince trees, and that's her quince on the left, and that's a store-bought regular commercial grade big quince on the right. And of course, the normal quince growers going, what the hell did you do? What fertilizers did you use? And of course, our answer was no fertilizers, just a healthy soil biome, having everything present. David Johnson, who we've now made an adjunct professor at Chico State University as well, in virgin desert soil, inoculated with extract from this inoculant that you can make in your own backyard, into this cover crop. You see on the left the 4 rows versus just planting the seed without it. And you see the difference in biomass. So what we know is that when we think in terms of moving away from an input consciousness, and that's what we used to do, bring the fertilizer, bring the pesticides, you know, bring the herbicides. Rather, if we go from a geochemical idea around soil, that it's just sort of mineral and we got to bring the elements, to a living system, a functional living system, then what happens is that the soil begins to respond. And what we mean in functional is that basically agriculturally we've broken the living biological system, and it's a broken system. And that's why we need so many inputs. And that's why we're degrading and tilling. And that's why we're adding and trying to push the plant. Whereas with the organisms, when they work in association with the plant, they begin to respond. The plant signals to the organism what it needs. The organisms, for their own food source, which is the root exudates from the plant, then begin to create or demineralize or move those nutrients. And so we're looking for a fungal to bacterial ratio into the 1-to-1 up to 4-to-1, more fungi than bacteria. Then you start to get quorum sensing organisms that come together to create things for the plant. You get a self-fertilization process. And we can capture up to 10 tons of carbon, not carbon dioxide, carbon per hectare, which is a magnitude higher than what most soil scientists are currently saying or predicting, which is a very exciting component there. I don't know now why it's going the other way. Crop productivity, here's something I learned in our research and work. We can actually improve photosynthesis efficiency, which I had no idea was possible, which basically means more carbon will be stored in the soil. You'll make it healthier. You'll feed the organisms more, and you'll take more out of the atmosphere and not release as much. You get an efficiency increase. You know, some Australians, he was a trucker and he wanted to get into farming in Western Australia with low rainfall. He started to grow barley. And what he did was he inoculated with a product just like David Johnson's, the Johnson Sioux bioreactor, only biology, no fertilizer. And he got double the yields of barley in his fields that his neighbors got. So he started to purchase land as his farming neighbors went broke and he was making a profit. So he didn't have fertilizer costs and he had double the yield. What this picture is interesting about is that there's some green in there and it's a very low rainfall. And you notice that green runs right down the row of the old barley plants that were already harvested. That is a perennial grass that has sprouted that has not been seen in the area for 40 years, and he didn't plant it. And what that tells you is when the biome is right, seeds will be stimulated and germination will occur. And for him, that's not a weed because he'll turn his sheep loose into this field in this off season. So he's getting another crop out of it as well, again, just with biology. The reason those two pictures are there with those roots that are all sticky with soil is because that means the biology is working. It means the roots of the plants are exudating, excreting, feeding the biology with sugars and carbohydrates, which are sticky, and the soil then sticks to it. If you pull a weed or a plant out of your soil and it's just the clean white root without the sticky substance, it's a quick test of it's not biologically active or functional. If you have what they like to call, this is a farmer I know for a long time, he likes to brag about his dreadlocks, you know, and that is versus the plant on the left that's white, clean roots, which has been fertilized. He's got all this biology working with the plant, freely fertilizing the system for those plants. So here's a study that was done unbeknownst to us in Belgium. They made their own Johnson Sioux bioreactors. They treated cover crop seed, and the blue graph is the cover crop that was inoculated versus the yield of biomass in the orange of the uninoculated. No fertilizer addition, just biology, and this complex biome that was re-inoculated into that soil. In Arizona, we have this 5-year project going on Howard Buffett's farm where we have a biologically enhanced, that's what we call it, where we inoculated the seed. And in the very first year, where most organic systems say you've got to take 3 years before you ever get yield back, we were able to nearly match the 250 pounds of nitrogen applied to the conventional system. Now that's a phenomenal thing to pull off. 126 pounds of nitrogen was lost to the environment in that conventionally farmed system. So if you're fertilizing trees or things in your backyard, or if you get a rain or you irrigate a little bit, a lot of it's leached away, pollutes the waterways, or it goes as nitrous oxide in the atmosphere as a greenhouse gas. If you make it biologically, if nature makes it for you biologically, you're not losing that to the system. The plant is utilizing that. So we increase profit to the farmer. The farmer walks out of the field and goes, why am I buying fertilizer? Well, all the fertilizer salesmen told you need to, but you don't, and you'll make a lot more money by not. We're just now planting our 3rd year of corn. I mean, it's our 2nd year of corn. Last year we planted beans. We'll be planting corn this year, and we're very excited to see the results of this project. And the pinto bean production in the off season, where we had the conventionally fertilized system, it was the lowest yielding versus the biologically enhanced plus 15% of the normal nitrogen applied was the best in the 2nd year. We think by the 4th year, our BEAM only, our biological enhanced, will probably outperform, but it outperformed the expense of fertilizer. How about that? 25 cents an acre cost versus $200 or I don't know what it is on the fertilizer cost. So that's where biology can do the work for us. And here's a picture that I want to leave as an impression in your mind to go to sleep with tonight. Here we know that, you know, when we get healthy soil and our roots grow out, we see lots of fibrous roots on many plants. But when the fungal network is engaged in the relationship with the plant, you can increase the root surface area by 50 times. So that means in water short years, that those fungi will bring water to the plant. If the plant is short of phosphorus, those fungi will demineralize the bound up phosphorus that your soil test said wasn't there, but is there, bound up in the mineral, demineralize it and bring it to the plant. If the plant signals, I'm short of one little molecule of boron, fungi will try and find it, bring it to the plant. And so it's where you don't need to, where I've heard some farmers say now, I don't get soil tests, I just feed my biology. And that's the kind of thing that we've got to pay attention to. What destroys these fungal networks? Why don't we have soils that are 4-to-1 fungal to bacterial? Because one, we till. In my work in Africa, we went to do some work in Burundi for Howard on one project, and every square inch of that country was tilled, every square inch. And that's just destroying this. The other way to destroy these fungal networks is to put fertilizer, put chemicals in the system. It will kill them. So we have to make the opportunity of stopping the tillage and stopping the chemicals. In my project in South Africa, my fields were phosphorus short by every soil test. My yields showed absolutely competitive numbers with chemically fertilized fields right next door. So what's that from? It's from the biology. We're doing a project with Metropolitan Water District on using this biology and cover crops to save water instead of going fallow. And we're very interested in what that study will reveal to all of us. We have this bioreactor consciousness starting to move around the world. This is my wife on the left standing with a group of Kenyan researchers that we visited in November of 19. They were just getting ready to do in a beautiful research center some research with this biology. This is a class I was teaching here in San Luis Obispo County. I think we have the highest number of bioreactors in the world. I hope we do. We have a lot of people building them, including public policy people, city council people, really thinking in terms of how we improve the county's soil health and carbon capture. So the source of my evolution in this understanding really of why we need to do this is we have a hunger problem, we have a drought problem, we have a climate problem, we have a water shortage problem, and that's all a soil carbon problem. With soil carbon increases, we get tremendous productivity out of the soil, we get more water holding and percolation capacity in the soil, and we suck it out of the atmosphere to correct our excesses of carbon release into the atmosphere. So these are the paradigm shifts that I wanted to share with you tonight. I have about another 4-hour lecture if you want to sit around. No, I'm kidding. I do, but I would say I'd love to respond to questions. The center is up in Chico, 6 hours away from me. We have 10 major research projects. We're doing certification courses now, starting to get some, will come online fairly soon. And we're doing lots of collaborations as the consciousness around regenerative agriculture is beginning to take hold. 61 countries are following our work. We have a bioreactor registration online. If you want to make one, just go to our website, which is here. It's listed here, I think, but just go to the Center for Regenerative Agriculture at Chico State, and you can make one in your own backyard. This is an inoculant. It's going to take 12 months for this biology to mature. It doesn't happen overnight. It's a no-turn system. There's no smell. There's no leaching. And you need redworms eventually in that system. So I'd love to respond to questions or thoughts any of you may have. So let me go out of share here in that show. and stop the share. So Ty, back to you. I rushed through that. I hope it wasn't too fast, but I'd be delighted to respond to any questions or thoughts.
Well, first of all, thank you so much. Definitely exciting to think about more people applying biology to their landscapes if they haven't actively done that yet. And yeah, thanks for your time. So we've got chats coming in. So there's one question about how an inoculation is done.
So that's a really good question. And so with this bioreactor, you end up making an aerobic compost. It's not anaerobic, it's an aerobic process. So the fungal communities can come forward. They eventually do very, very broadly at the end. What I mean broadly, a wide spectrum of them. So how do you inoculate? You can take some of that compost and put it around your seed, but an easy way to do that, if you're going to plant in a garden, if you're going to plant a bunch, is to simply take a bunch out, stir it in a bucket rapidly to shake the organisms loose of the organic material, dip your seeds in it, and then plant your seeds. That way the seed is inoculated. When the root hair comes out, the organisms, like a birth canal, the root hair and organisms start to work together. And the relationship begins at birth, so to speak. So everything starts to function really well. If you are planting a tree, I understand there's even some nurseries or whatever up in the Midwest that they're starting to guarantee that if you buy a bare root from us with this mycorrhizae fungi, we guarantee it will live. Well, they're getting the idea about fungi. We like to do the whole broad spectrum, but mycorrhizae is a core element to this. I mean, it's one of the real keystones of this whole system. So we don't argue with that. It's just that you can do this in your own backyard, or you can buy mycorrhizae and try and get a broad spectrum, get vermicompost, whatever. You can add a life to your soil in other ways. But what happens with a lot of anaerobic compost is you're not high in fungi, you're high in bacteria. And that's what we're trying to shift, is that ratio. Put your compost in, but if you can get this inoculant in, you're going to bring the fungal communities with you, which are really important.
So that was partnering. There's another question partnering in with that, which is asking if regular compost does the same. I guess we'd have to define what regular compost would be, whether that's homemade or purchased in a bag.
I would probably feel better about the compost you make at home than what I purchase in a bag. But in fact, I just made the point that normally your regular compost at home is turned. It is typically anaerobic. Sometimes, you're putting food waste and you're putting green stuff in there and your temperatures go high. And what we're doing in the bioreactor is you're putting brown material in. You're not really putting green material in. You don't put food waste in, and you don't jack that temperature up real high. It's gonna get some warmth to it, but after a couple of weeks, you put redworms in and you just keep it wet. Keep it at 70% moisture and don't touch it. So that way the fungal communities will come into that system. So they're very different outcomes, and it takes 12 months on this one. It takes a long time. So here in San Luis Obispo, the rare fruit tree growers went out and made one at their joint orchard, so that everybody could see it and that everybody could access some, because really it's only going to take a big handful for most people's backyards if you're going to inoculate things. If you can do 350 acres with a full-size bioreactor. So I'm actually making smaller units than what are on our website, and I'm trying them out and they seem to be working fine. We have some students at Chico Mechanical Engineers that are developing an even smaller one and running trials on it. And they may even try and market it, which is great if people don't want to go find their own materials. But you'll have plenty if you make one for your yard, or even a number of acres if you have that many trees.
Great. To piggyback on that, I have experience looking through a microscope at soils and composts, and it's amazing how I had a disconnect of the quantity of biology that can be in the tiniest amount of compost. And so it's surprising when we're used to maybe buying a dozen bags of a compost or thing from the store to inoculate or to cover the soil with or fertilize a bed. When it's a biological focus and it's homemade stuff, it can go, little can go a really long way, to echo what you're saying.
And it's a good, Ty, that's a good point, but also a lot of our compost, we're adding carbon to our soil. With the inoculant, you're going to create it for your soil. The plant is, because it's going to exudate so much carbon to feed. When you have a healthy biological soil, up to 50% of your carbon in that soil can be the bodies of the organisms that have died or been partially eaten. So that's not leaf matter or roots, that's actually biology. So it is a very different process. I would not say don't put compost in.
Right.
I'm just saying this is the element that brings the fungal communities and does a direct relationship with the roots, whereas a lot of your compost are just bacterial dominant, but you're bringing humates and you're bringing carbon to the system. And that's good. There's nothing bad. That's why you see a response. You're bringing a certain amount of nitrogen, which actually I'd rather have that all be created by the biology. And that's a long conversation, but it can be, because that means they're working, biology and plant are working together all the time. To keep that biology alive, it's great. And those of you into trees, this is perfect. It's best to have a live root in the ground 12 months a year. Anytime you see bare soil agriculture out there, you have soil going to sleep or dying because it's the organisms are not being fed and they're not being kept alive. So that's where one of agriculture's big flaws in its understanding of soil and why we're trying to make that change. There's a question here I'm seeing that says, is this bioreactor best in full sun or shade? You want to keep it moist. So a lot of people make covers for it, and you set up a little timer irrigation system and put a few minutes spray on it every day. So thank you for the webpage, whoever put that up. Thank you.
That's Robert. He's quick to the draw on providing.
Thank you, Robert.
Thank you. So also in there, fitting in with what you're just talking about, there's a question on if organic fertilizers would destroy fungal networks.
Organic fertilizers, probably not, but they're going to dissuade. What'll happen is it depends what the organic fertilizer is. So you can get organic fertilizers that, if it's more directly towards nitrogen, that stops the working relationship. So if you get an IV and get your nutrients through an IV, you don't need them, to work for them from the standpoint of eating, digesting, and processing it in, feeding your organisms in your gut. The plant and the organisms, because that root not only will exudate and feed the organisms will bring 5 different kinds of nitrogen to the plant. So there's different biological resources for the plant to get its nitrogen. The other thing that I didn't even know about until a few years ago, and there's a scientist out of Rutgers that showed that plants actually consume bacteria. I never knew that. I always thought that they only lived in the rhizosphere and there was some exchange, but they'll consume them and take them in, and in some, they'll spit some of them back out still whole, and those bacteria will still repeat, but they're actually consuming some of the protein, therefore some of the nitrogen, therefore some of the minerals. So the healthier that rhizosphere is, which means the organisms around the root, on the root, the more present, then the healthier the whole system is. They're working together. And it's sort of like, if you work out and eat well, you're healthier than if you sit and just take your nutrients in through an IV. It's a very, very different, being. A healthy, immune, resistant, strong one that's going to resist actually insects more, because the Brix level in the leaves are going to be higher. The insects are less attracted to it. And the fruit should taste better. And you'll get those huge quinces, I promise you. So try it.
Who doesn't want bigger quinces? Come on.
That's right.
So another question, I'm getting some dog vomit fungus in the vegetable garden. Is that a good thing? It's the fungus that often sprays spores up, I believe, when water hits it. Is that the same one?
Yeah. I am not a fungal expert, but I'll tell you one of the things that happens is that anything that is out of balance, it probably means there's something out of balance. So I don't know that there are two kinds of funguses too, because there's the ones that feed and build, and there's the ones that have a very appropriate place in the ecosystem that consume and degrade and take apart. And so we need them all. They all have to be there. And that's why we can have, to say, I have E. coli in my gut. You go, is that a good thing or a bad thing? Well, if it's out of balance, I'm sicker than a dog and may die. If it's in balance, I'm healthy. And so my wife had given a talk last time we were in Rwanda. I was giving a talk on this subject, and she was giving one on aflatoxins, and she did some research and found because she's from the healthcare world, and she had found a study done at North Carolina State University where they did the same kind of composting, and where you had a complexity of the fungal communities, aflatoxin, which is a fungi, which is toxic to humans, was still present, but not at a level that would do any harm to anybody when the complex system was there. When the complex system wasn't there, it would get out of balance, and then it was toxic and harmful. So all I would say is on that, if you see it in your garden, I'd watch. I would, if I needed to try and make an amendment, I'd try and do it biologically. And I'd have to read and study about that. I don't know about that one.
I would say I've seen it in our own yard, seen it in plenty of others. Usually it's present when there's mulch, looks kind of like a dog threw up. And I think it might be a decomposer of woody material. If anyone wants to reference more on fungi and fungal ecology, we do have a presentation that was given last year by Peter McCoy of Radical Mycology, and we have it unlisted on YouTube, so it's not searchable at Peter's request, but we do have that available to chapter members if anyone wants the deeper dive. I was actually just watching that today. Okay. Is there a specific type of mycorrhiza that's recommended? What's that?
No. I was gonna say, I see a question here that says, can we see pictures? Yeah, they're on the website. There's like 250 landing pages on that. There's so much that's happened to us in 5 years, so much work, so much interest. But if you go to the bioreactor registration site, the David Johnson site, there's a film on how you build one, how you inoculate, all of those questions. So there are pictures there if you want to see them. I'm sorry, you had a comment, Ty?
There were more questions. I was just kind of starting at the top and moving down. So is there a specific type of mycorrhiza that's recommended? There's so many different brands out there.
Arbscular mycorrhizae is the primary one. There's an endophyte and non, and the one that we're looking for is called AR mycorrhizae, and that's generally the one for most plants. Now some trees use different kinds than what some agricultural crops use. And that's why I say, make this in your own backyard. And all we're saying is when you make this with brown material, leaves or straw or whatever, and you put it through your lawnmower or grinder or something, so it's not big corn stalks or something in there. So the worms and everybody has a chance to decay at the organisms over time. You get the whole array of fungi and bacteria. You'll get thousands of species of bacteria. And what happens when that gets inoculated in the soil, depending on the tree or the shrub or the agricultural crop you plant, it becomes a self-organizing system. So what the plant signals for and starts to feed, those are the organisms that come and start to reproduce and work in relationship with that plant. And that's why the broad spectrum, it's not that you have to go out and buy a specific X, Y, or Z. It'll all be there because it all lives in the air, lives in the dust. Spores move, it'll end up in your compost, and it will then, because it's in the proper culture, it will develop.
Okay. This sounds like organic gardening. How do you differ?
Organic gardening often is high input gardening. It is no different than, most of it is no different than chemical farming because it's input consciousness. And this is where this is an organic process, but the organic world doesn't understand this, just like the conventional world doesn't understand this yet very well. We're learning every day more about it. But what this does is this is a no input system. Once you've inoculated and gotten your soil very robust, if you look at a tropical rainforest, who's putting nitrogen on that dark green forest? The biology's doing it. The same thing can happen in your own system. Once that soil is very, very inoculated and the roots, and you have diversity within the system, and you keep your soil covered, hopefully with live roots. Second best would be with mulch, but live roots are the best. Then you really are not bringing inputs in, but organic often needs compost or fish emulsion or something as it's trying to bolster also a soil that might be about as dead as a conventional chemical soil that's not been tilled, but the organic has been tilled, because that's the only way organic usually controls weeds. So this is where it steps beyond that into making a functional living system that becomes self-organizing, self-fertilizing, and can be very, very productive on net primary productivity.
All right. There's a reference here. I don't know if this is a question, but it's referencing the groundbreaking work of Suzanne Simard regarding soil bioactivity in forests. It's just a note.
I have her book right over there, because she was talking about the mother tree. And I think that in forests, here's the interesting thing. In disturbed soil, in our backyards, if you see a lot that's cleared, what grows there? Weeds grow there. If you go out to agricultural soils that have been plowed and dished, or our garden that we keep hoeing and et cetera, what do we see? A whole bunch of weeds. Well, weeds do best in a bacterial dominant soil. And some of those soils can be 100 to 1, what are bacteria to fungi? When you get to a 4 to 1, fungi to bacteria, agricultural crops do the best. When you get to a higher fungal level, trees and shrubs do better. And what is an old growth forest? It's about 1,000 to 1 fungal to bacterial. Do you see weeds in a forest? Not if it's undisturbed. You see trees and the understories that grow there, but not weeds, because it's a mature soil that's highly fungal dominant. We can't grow crops in there anymore. I mean, we have to disturb it to be able to grow crops and get rid of a lot of that. That's too much fungus for most of our agricultural crops. But we have soils that are almost bereft of fungi in our fields in California or Illinois or Africa, because we've killed them.
There's a question on if it's possible to speed up the process. You mentioned 1 year for the Johnson Sioux.
When you discover that, please do what we're doing, open source it and tell us how you did it.
But maybe they can purchase it from someone who's made it. I guess that's a little faster.
We're trying to study, we don't have the time, resources, or money. We're trying to study, are there marker species in there that can tell us that you actually succeeded and you got this fungal dominant compost at the end that you can inoculate with? And still, it's either a very expensive process, or you can get there's a couple of tests you can get for about $20 to $40 from certain labs, but it's hard to know. And people can fail at it, and you can go, gee, it didn't work. But then we may find out that actually it didn't stay wet enough, or you were in Minnesota and you let it freeze like a popsicle. We don't know all of that yet. This is a very new science, so we're just learning.
A question that maybe on speeding up, this is a question for myself on speeding up the process of sourcing it from someone who's already made it. Is it region specific, or like someone making compost on the Central Coast in this manner effective for down here?
So one study that a local friend of mine here she helped fund it, and she's going to start to try and make it on a commercial level so it can be sourced by farmers, by anybody, because the planet needs it badly. If we can capture 10 tons of carbon per hectare, we can fix this climate thing by converting our ag lands to a fungal dominant soil, a very healthy fungal dominant soil. But in essence, in that study, David Johnson, he got 10 samples from across the country, from New York to California. Mine was in there, his was in there. And serendipitously, he got the same genomic analysis from 4 samples out of Australia, made from very different materials, but still brown materials. And what happened is the Australian samples matched his, which we know is the best compost that he made. The rest of us didn't quite match his, but it's more to do with we weren't as attentive to taking care of our bioreactors as he was and the Australians were. And so that'll show you, no, it's not regionally. You could use City Farm here in San Luis Obispo. They have a mature bioreactor. I want to go get some of theirs. I have 4 bioreactors here myself, but I'm a few months away from being able to harvest anymore because I gave all my old stuff away. But in essence, there are people that are going to make it, that are already making it, that if you can find a little bit of a source to get started or run your trials or do some greenhouse pots with it, or, you know, do some roots of your trees you're transplanting. There are some people that have some that you can get, but it's going to take us 12 months, each of us, to get it ready. And then we can start to share.
Right on. There's a question on the initial things that you recommend that someone do if they're interested in going down this biological route. Build a bioreactor, is that the starting point?
Build a bioreactor, but there's some principles. We've got to figure out how to stop tilling so much. So any tillage you do it very, very, very minimal. So you need to pull a few weeds or whatever, but don't sell your rototiller. That's the worst thing in the world, or your disc or whatever. Those are terrible. Second thing is that you want to keep a live root in the ground all year long if you can. So if there's water shortages, or, you know, what am I going to do? Mow that? I don't have sheep to graze it down. What do you want me to do with that? The second best thing would be mulch, but that's not feeding the organisms. It's just keeping your ground cooler, and it's feeding the earthworms. And apparently the dog vomit fungi. But anyway, you want to, you know, keep that soil covered. But the best is to have live roots in the ground. So if you can plant other things under your trees, you know, get diversity in the system, because diversity below ground is reflected by diversity above ground, or vice versa. So you're going to feed different organisms with different roots and different plants. And you're going to host more insects and beneficials with a wide variety of pollinating plants. So I would say those are all regenerative biological feeding approaches that's not destroying it. And then reduce or eliminate chemicals. So be very, very judicious with chemicals, fertilizers or herbicides. That all kills biology. You know, it's sort of like you're trying to grow something and then you're throwing sides in there, fungicides, you know, herbicides, pesticides. So that's called suicide. So you don't, as little of that as you can.
All right. Someone had been reading about actively aerated worm tea. They read that by using different time periods and pH, you can tailor the bacterial fungal balance. Is this a useful pathway?
So I know somebody that's doing that, and I think it's a great product. It only lives in a jar very well for a day. So, you know, this is why I say make this stuff in your own backyard, make your own extract, put it through a paint filter if you want to do that, and take your yard sprayer and spray it. Be sure you've gotten all your chemicals out of your sprayer. Spray it on your seed, or spray it on the roots, or dip your roots in a bucket or your seeds in a bucket and then plant. But you can buy product, but a tea that's been made with complexity and with fungal spores in it, as long as the fungal spores are there, I couldn't complain about. Particularly vermicompost is going to have more diversity than general other systems.
Right on. You had mentioned the inclusion of red worms towards the end of the process. What role do they have in biodiversity?
I don't know. They just have not been that forthcoming with telling us what the hell they're doing in there, but they absolutely, after the first few weeks you throw them in, they are just part of this composting process. And so it's not exactly like a vermicompost system, like if you had your worm bin. And I'd use all of that product in my soil. It's good because it's biologically alive. It's not just nutrients, it's biologically alive. But they have a similar system in this. It's just that this guarantees a heavy fungal community at the end. And they either don't interfere or they help it, which nobody's revealed that secret yet, which as a scientist today, I don't really care. I think nature can hold some secrets. We're just looking for results right now. how can we build and support the living system? And they seem to be an important part of this process in the bioreactor.
The question is, has there been research on placing the bioreactor ingredients in situ, effectively turning the soil surface into the bioreactor?
I don't know if I fully understand that. Yeah.
I think the idea being not having a structural bioreactor, but being able to do it in place in the landscape, like across the landscape.
No, because you have to keep this very moist. You have to keep the moisture, and you've got to get air through it. So on the land is it's going to air out and dry out typically. I mean, you're not going to have enough water to keep it wet enough there. This upright system is what seems to be crucial to let air flow, and then water to keep it wet. That's an important. We kind of think about it. It probably would have been developed in a agroforestry system, or I would say a semi-forest system, not a heavy old growth, where you would have some shade, moisture, leaf drop, get fungal communities and bacterial happening at the same time. So we're sort of probably replicating nature in that way, but this is getting the air through it. So let's get it lifted up vertically, and then keeping moisture on it and not turning it.
Well, it's definitely going to be ultra dense in terms of biological diversity and quantity. And like you said, can spread across a wide area for inoculation. But I can imagine, you know, any mulch and or root stimulation, if there's some biology present, there's going to be feeding and replicating and spreading. But this is kind of fast tracking in a slow way, and also giving you the option to spread it across that whole space. Okay. Does biochar have a place in this system?
So biochar is dead carbon. People like to say it's a hotel for organisms. Well, it's only gonna be a hotel if you inoculate them. But and I'm not against biochar, I'm for it, except where's the feedstock coming from? Don't take your leaf matter off the soil and take it to a biochar. Leave the leaf matter on the floor to feed the biology. If you're taking walnut shells or peach pits, go ahead and biochar those, that's fine. But in essence, my thought is always, why would an organism go to a hotel? Why would it leave the buffet of the rhizosphere, which is where it's getting fed, and check into a hotel? So I don't understand. I think it'll add carbon, but it's kind of a dead carbon. Why did the terra preta soils work so well? I don't know, but I really don't understand all of that. But I do know that we can capture more. We're in a living carbon cycle we're working with. Biochar, I think, is great. I wouldn't spend money on it. And there's very little like biochar. And I did my own experiments with it in Africa. And the people there needed the wood for their own home fires, either to heat their homes or to cook with. And so I kept thinking, what would we biochar here? And the crop residues needed to go back on the soil. So it became a moot point for me. And here I see, with all of our nuts grown or pits, we could biochar that as an energy source. And then it's sort of a byproduct that if we inoculated it, I think it would have some value for the soil. It does put hard carbon back in the soil, so that's no longer in the atmosphere. And that's a good thing.
Well, there's definitely a lot of waste on the planet, so of biological remnants. So I'm sure there's places everywhere to source that type of material, but just not in every context, like you said. So there was a question on, is this applied as a tea? I think you kind of addressed that. You can broadcast it, right? Or do a water extract. And that's not, you're not necessarily recommending with air, like a active aeration and food, like an aerated compost tea.
I'm talking about a tea. This is not a compost tea. It's just
right.
an extract from your compost. So,
extracts are not over time with air and food, right? That would be tea. Okay. So someone's commenting, actually question, biodynamic agriculture sounds similar to what you're explaining. Is that so?
I really think so. And it's curious because that compost is no turn, but it's not upright. But there's a lot of earthworms that get involved in that. If you have a living soil, they're just by accident or by opportunity. I think so. I would love to do studies on what biodynamic compost, good piles, what they produce, how much fungal matter is in there. It's very alive biologically, for sure.
Are there any books that you recommend?
Not yet. I mean, there's the biological stuff. The books I recommend basically are Regenerative Agriculture, like Gabe Brown's book, From Dirt to Soil, or Nicole Masters' book, For the Love of Soil. These are very readable, and they're not particularly talking about David Johnson, but they're bringing the biological conversation in. They're very regenerative, which is building this whole living system.
I'm forgetting the one. There was like a 3-part series a guy did that's very popular. One on fungi, one on biology in the soil in general. I'll pull it up in a second. That's a great primer, and it goes into a lot of depth on nutrient cycling and how it's very like detailed on the biology. I'll pull that up in a sec. So someone has a compost bin that's 10 foot long by 3 foot wide by 2.5 feet deep. If they put tubes for air, would it accomplish the same as a bioreactor? I do not turn it, and one end I put a lot of brown waste. I put a lot of brown waste around a year ago.
Possibly. The biggest concern for me is the bin is probably not permeable for air movement from the outside. So if you get enough air through, David Johnson always says air needs to be accessible to the material no more than 12 inches apart. So if you got enough tubes running through it, it possibly could. That would be an experiment, maybe worth trying, but I can't tell you because we have a fabric that we recommend that lets air move from the outside, and tubes inside that lets air move from the inside. So it's just easier in my mind, but innovation is always worthwhile. So it might be worth exploring.
Okay. A question, this person doesn't necessarily know the whole process, but is it explained when it's actually ready to harvest?
There's a texture that it looks like, but and your pile is going to start if you literally have the size David Johnson makes, it's over 5 feet tall, and when you're done, it's about 1.5 feet tall, and so it's really composted way down, and you can rub it through your fingers. And it's like clay. I mean, the moisture is high, and that product is like a clay, like your real clay soil coming out, and that's when you kind of know it's processed through. Unless you did a test to see what the fungal, the bacterial is, I wouldn't be able to tell you, did you actually fully succeed? But probably, if it ends up in a product like that.
Okay, and what keeps your microorganisms alive when you add them to the soil?
Living roots, because they feed them. They feed them the sugars and carbohydrates. It's the relationship. So that feeds them. That's their energy source. That's their carbon source. That's why it comes out of the atmosphere photosynthetically down through the plant, and the plant, because it needs the nutrients from those organisms, feeds them, and it exudates. I didn't show you the little root here where it's spewing out all these sugars and carbohydrates, feeding, because it needs what the organisms bring back to it. So that's what keeps them alive. Okay. Soil really is the plant, the mineral element, and the living biology. And that's a living functional soil. If any one of those is gone, it's not that, it's dirt.
John, I'm seeing your question. Do you want to clarify that on air being forced through the soil?
Yeah, you were Tim, you're talking about if somebody came up with an idea of speeding up the reaction. I'm thinking chemistry again. I'm used to fluid beds and things like that, where you force air in through a medium so that the air is actually permeating through the product that you're going to dip, say, a hot metal frame into and coat, like you would do with a polymer coating. And I'm just wondering whether you've got static pipes and you've got air coming down through the pipes and then it's permeating out that way. What if you were able to force the air through the soil? Would that work faster?
Well, because it's a biological, not chemical function, I don't know. And I don't know that it would or wouldn't. So what's happening is that you need to keep the moisture level up. So if you're pushing more air, you're gonna have to push more water, and that's all right to give that a try. But does that then enhance the speed with maturity? And so if you look at on our website, one of David Johnson's full presentations, about an hour long, he will show some graphs of the metagenomic change in this bioreactor over time. And the number of species starts to really magnify right at the end. And so can that process be sped up? I don't know, John. And that would be something, if somebody with time and money on their hands, they could run and try and look at the metagenomics on it, on what's happening with added air or with added, this or that. Don't know.
Right.
The question.
A little bit.
What's that, John?
A little bit of, the biomass goes a long way as far as spreading the microorganisms.
Yes, very little. Yeah. Yeah.
All right, I'm seeing the end here of the questions. Got a couple more. Do the beneficial fungi only have a beneficial effect on the roots, or can they have a protective effect when sprayed onto the leaves and wood?
Yes, absolutely. So a extract that you spray on the leaves, just like on that tree, it looks like it can really assist the plant too, because remember I said about humans, we have a biome of the gut, a biome of the skin, and a biome of really our lungs. It's the same on the plant. And there's a biome on the leaf and on the outside of the plant. And so it absolutely, we've seen some people testimonially talk about it. We haven't done trials or studies on it yet, but it makes sense to me. So I would absolutely do that. We think it could probably affect a lot of plant diseases in giving resistance to that. And as the Brix level of the leaves grow with your soil health growing, then you're going to get it from both directions, the external biome and more nutrients to the leaves and sugars up into the leaf too.
Someone's asking if they're understanding the directions correctly, that once the bioreactor is filled up, you remove the 6 PVC pipes.
Isn't that amazing? I know, and I don't even like to do it on my own. But that's what David says. He says after a day or two, he just pulls the pipes out. Well, that lets more air in that shaft rather than what the pipes with the holes would give. So, and it's easier to do early rather than later usually. What'll eventually happen, which I can't for the life of me figure out why this still works, eventually those holes will collapse, but not for a long time. So a lot of the process is done, I guess, by the last few months of that system. So you won't see the holes at the end, but they'll stay intact within about the second day. You can just pull them straight out.
One of the books, Martha hit it on the head, is called Teeming with Microbes. Have you heard of that one? Jeff Lowenfels. Yeah, it's a good primer on microbiology and specific to organic gardening. Well, gardening with biology. Okay. I guess, John, you had another one. Has this process been scaled down to fit in a 5-gallon pail, or is it a mass-based process that needs a critical mass?
I think it needs a critical mass. But John, one of the things that I did because I cared so much about Africa and small-scale farmers, smallholder farmers, is I made my own African model, thinking in terms of no cost kind of construction, because they don't have resources to build them, and they have a hectare or 2 hectares or 3 hectares they're farming. And so they need very little. And I just used some scrap horse fencing wire. I took peach tree prunings to make my floor to get it up off the ground. And then I used a seed bag, which most of them have because they either can get them or they bought seed to plant in their fields. And so it was only about 18 inches in diameter, and it was about 3 foot tall. So a much smaller scale. And it looked like I put the worms in, it looked like it was just doing great until about 10 months. And my seed bag deteriorated and fell apart to pieces. So my system fell apart, and then I had all these terrible plastic particles, particulate in my compost. But I think in terms of African, most villages would have a basket weaver, and I just think they could make it out of reeds and probably use it for a couple of years, couple of different seasons, and still get some airflow and make it rigid enough and any size they want, but keep it wet and have enough product in there for the worms to do their activity over that 12 months. So you'd want some scale to it. You don't want a tiny little container, so that there's enough activity, enough material, and enough chance for the fungal communities to get started.
Well, that looks like all the questions.
Thank you.
Thank you, Tim. Yeah, super generous with your time. Thanks for joining us. And I think that's all. John, you got anything? Tim, any final?
Thank you, Tim. It was enlightening. I know that there's a lot of mycology work being done at the moment. It's still not a topic that I see in universities or part of soil management, things like that, and I know it should be. I know the Australian Aborigines knew the value of creating good soil, and before white man came, all of the plains were like meadows, and of course the white man said, "Oh, this is great land for grazing," and within 2 generations they had destroyed all of that pastoral land because of the excessive grazing. But I find it a fascinating subject, and I hope we can do something here to spread the word and to help change the mindset and the culture that's out there. And even if we start off small in backyards, or even if we have a collective somewhere that we can make one of these bio feed systems that people can draw from, I think it'd be a good project. I know your chapter up in the Central Valley did this, and they talked about it in their recent newsletter. And I've circulated this to the board, and I'll have to ask for approval to go beyond that. So it's of interest, and we have a propagation committee, and if we do plant growing for our plant sales, why not dip the roots into a medium that helps promote growth and microorganisms? So I see some real potential if we could get all of these CRFG chapters to promote this sort of thing, to start off with in backyards and then go beyond that.
Wonderful. That would be marvelous. Thank you very much. And thank you for being open to a new paradigm and investigating. And it really promises a future for the whole planet, for the health of our food and our gut. As I always say, if I'm working in my garden, I don't bother to wash my hands before I eat. If I get off an airplane, I go straight to washing my hands, right? I mean, it's what organisms do we want in there? What organisms do we want to avoid? And blessings to all of you. Have a great bioreactor exploration, and thank you for being a part of this conversation tonight.
Okay.
Thank you, Tim.
Thank you very much, Tim. I really appreciate it.
Thank you. Bye-bye.
Bye.
Take care.